Hi, everybody. Today we're going to look at biochemistry, and we're going to start with the energy molecules in biochemistry. So we're going to split biochemicals up into 3 different videos, and this is the first. The first thing I'd like to talk to you about, though, is polymers and monomers. That word poly means many. The mer refers to a part. So a polymer is a molecule that has many parts to it. Mono, of course, means one. Mer means part. So a monomer is one subunit, like a link in a chain, and a polymer means you have a chain of many smaller subunits. All of the biochemicals that we're going to be talking about are polymers. So they are all made of repeating segments of smaller molecules. They're put together by a reaction called dehydration synthesis. This is also known as a condensation reaction. Notice you're pulling water off. In other words, you're making water, which is what makes it a condensation reaction. Essentially, what you're doing is you're joining the molecules covalently and making a water in the process. When you take them apart, you do a process called hydrolysis, which literally means water splitting. Okay, so when you want to break down a polymer into its monomers, that's how you do it. This is what happens when you digest your food. All right, so let's look at this condensation reaction or this dehydration reaction. So here's a polymer that's short. Here's an unlinked monomer, and I'd like to add this to my polymer chain. There's a hydrogen sticking off here, a hydroxide sticking here. If these two are pulled up and water is removed, we have the availability to make a bond, and that's what you're seeing here now. The polymer is longer. In order to undo that, to digest this molecule, all you have to do is put the water molecule back in, and now you've broken that up into the short polymer and the monomer that you've pulled off. All right, so all biochemical families exist as polymers in one sort or another. Well, sort of. We'll get to that in a sec. Carbohydrates are the first of the energy molecules. These are the sugars and starches. Now, sugars are mono or disaccharides. So monosaccharides contain one monomer. Disaccharides contain two monomers, and then disaccharides or polysaccharides would contain many monomers. So some examples here obviously are glucose, that's a monomer. Sucrose is a disaccharide. What we get when we join these together is a little bit of a different kind of linkage. It's referred to as glycosidic linkage, and essentially what it is an oxygen that's holding the two monomers together, and I'll show you that in a minute. So it's not that the carbons are bonded directly like we normally think of in a polymer, where one carbon in one monomer is bonded to another carbon in another. Here you've got oxygen kind of holding on to the two monomers. Carbohydrates are for energy, but they're used for short-term energy storage. So sugars generally get burned off. If you don't burn them off, then you would store them as starches, and starches are the polysaccharides. Not all polysaccharides are used for energy, and we'll look at a few of those in just a minute, but these have hundreds or thousands of monomers joined together, and usually they're energy storage. But again, this is a shorter term energy storage. This is found in your muscles. This is the kind of starch that you might find sometimes that plants would store, like amylose, for energy, and then they can hydrolyze these starches to create the sugars that they need to fuel themselves. Occasionally, you will find polysaccharides for structure. So they're not starches that are used for energy, they're structural molecules. And that's things like cellulose in plants, which is what the cell wall of the plant is made of. All right, so animals use glycogen in muscles for energy, chitin for structure, and plants use cellulose for structure, and they usually store energy as amylose and amylopectin. Okay, and again, the difference in these is the types of glycosidic linkage between the monomers, but also the degree of branching. So let's look at some pictures of what this looks like. All right, so here are some sugars, right? Sugars can take on a chain structure like you see here, or they can actually kind of fold back on themselves and create what's called a ring structure. Okay, and this is what the ring structures look like, these little hexagonal or pentagonal structures. These are hexagons, some sugars are pentagonal. But notice here's amylose. Amylose is a very, very simple kind of starch in that it's just a chain, a straight up chain of molecules. Amylopectin has some branches. So here's one branch, here's another, and then it goes into a main chain. Let me see if I can move this. Oops, I was going to try to move myself over. But glycogen, this is the glycogen molecule. It's a slightly branched chain as well that's found in animals. And here is cellulose. Now, cellulose, the reason why cellulose is for structure and not for energy is the way that it is arranged into these fibers. And these fibers have cross linkages with adjacent fibers. So these are very, very complex molecules. They've got lots of cross linkages, and that makes them very difficult to break down. Chitin. This is what a chitin monomer looks like. This is part of the exoskeleton of arthropods. And chitin would be also a structural polysaccharide. All right, now the other kind of energy molecule that we have are the lipids. And these generally, if you're talking about fats, these are long-term energy storage molecules. So these are meant to be very, very stable molecules that the body is going to use primarily for stored energy for a rainy day. Not we're going to use it tomorrow, or we're going to put this in the muscles for a time where we need a short burst of energy. This is we put energy in lipids for long-term storage. This is why fats are so hard to break down. They're chemically so very stable, and it takes a lot of energy to break them down, as anybody who's ever tried to lose fat on their frame will tell you. Now, this also includes the oils and waxes. There are a lot of sterols that are lipids, like cholesterol is a sterol. That's a lipid. These are all nonpolar. The exceptions are phospholipids, which we'll get to those in a later presentation. But where the carbohydrates are like your energy checking account, these are your energy savings account. 1 gram of fat stores 2 times more energy than a gram of polysaccharide. The typical structure you're going to see, though, the one that you need to know, is actually not a true polymer because you don't have these tiny little molecules that are arranged in a row like you would see in your typical polymer. A triglyceride is an E-shaped molecule that contains a backbone of glycerol with 3 fatty acids coming off of it, and I have a picture of that coming up. This is the main component of the lipids are the main component of phospholipids. Recall that phospholipids make up the cell membrane. We're going to get into, in our modeling activities, the difference between saturated and unsaturated molecules. But essentially, when you're looking on the main hydrocarbon chain, are you seeing double bonds or single bonds, right? If you have all single bonds on the main hydrocarbon chain, then it's saturated. If there are any double or triple bonds, then it's unsaturated. And then, of course, trans fats are unsaturated fats that started their life as unsaturated fats, and then they had their double bonds broken and hydrogen pumped into them to create a saturated molecule. You find trans fats a lot in processed foods because they're shelf stable, but everybody knows that the saturated fats are less healthy for you. So a lot of times companies will make trans fats so that they can say that a product is made from unsaturated fat, even though it itself, the product, is saturated. All right. So here's what I'm talking about. Here is a triglyceride. This is our E-shaped molecule. Here's our glycerol backbone, and then there are 3 fatty acids coming off of that molecule. Notice there is a double bond here, but this is a functional group. Okay, so this is a carbonyl functional group. that denotes that this is a fatty acid. It's an organic acid. So we don't count this double bond as part of the main carbon chain. We're talking about the chain of carbons, and at each of these points, there's a carbon here. When you have saturated fats, like I said, the saturation makes them really, really super stable, chemically stable. When you introduce a double bond and you create an unsaturated fat, as you're seeing here, what happens is you introduce a kink into the molecule. And this is a point where now we're forcing a bunch of electrons, actually two pairs of electrons, into a very, very small space here. And that makes this less stable. It makes it more likely that this bond is going to break and that hydrogen or sometimes other things will sneak in. This is why we store olive oil in green bottles or dark colored bottles, because even the light in your kitchen can break this double bond, and then stinky molecules like sulfur can get in and make the oil go rancid. So typically saturated fats are solid at room temperature. Unsaturated fats are liquid at room temperature. That's kind of how you can tell the difference when you're looking at them. But otherwise, if you see a kink in a fat, you know it's an unsaturated.